Okay, so here's where we are in the outline. We are in part 2 here, so we're going to talk about transport epithelia. So we're still talking about osmoregulation. I'd like to, and then the next part of the lecture is going to be on nitrogenous waste removal. So we'll be going into excretion after that. So hold on. So we'll look down here at what we call transport epithelia. So this is an example of cell types that will, or a situation where cells are going to help via the bloodstream get rid of salts that may have accumulated in birds that are like marine or seabirds. This is an albatross. So what is happening here is, so if you look at the situation, so they have a gland in their nasal cavities. So you can kind of see it there on the top of the bird's head. And in that gland are cells that are tightly knit. They're going to be held together by tight junctions, so it's really hard to get between the cells. The only way to get anything through there is to go through an actual individual cell. These are going to be in tight association with the bloodstream. So right here you can see the situation where you're going to have blood that is coming in, which is the red, and blood that is going back, which is the blue. So the blood that is coming in will, say, for example, contain a lot of salts. And it is going to be passing past these, what they call the transport epithelia, which are the cells that are lining this gland. And this gland is going to be picking up salts and taking it and excreting it out of the nostril of the bird. Okay, so what happens there is that the blood, it has a high amount of salt, and inside the duct itself over here, where the blood has a high amount of salt, it will easily move into the cell by solute concentration gradient different. And then it will go into the lumen of the secretory tubule. And so it's going to move into there. And where there's a high amount of salt, there will end up being a high amount of salt down here. And then likewise up here, as the blood moves past, it's getting less salt in it. But still up here, there's less salt up there because the duct movement is in this direction. So since it's moving in this direction, there's always less salt up here. So regardless of the fact that this has lost salt, it's still going to be a higher solute concentration right here versus right here. So the salt will tend to move into the duct. So this is kind of like a counter-current sort of situation. That means that at the top part of the duct up here, there's a less solute concentration. And as the blood flows in from the bottom, it will always have a gradient into the duct, and therefore the salt will end up in here and can be excreted out the nostril. Okay, so that's transport epithelia. Okay, now we're going to move into talking about nitrogenous waste removal. So this is basically the excretory systems or excretory process that we've been talking about in other animal groups, and we'll kind of go through those groups again and sort of remind you of what that is, but not right now. For now, let's just talk about the different kinds of waste that can be removed. So this is a figure in your textbook. This is talking about the different kinds of waste. Of course, nitrogenous waste come mainly from proteins and nucleic acids. Nucleic acid, of course, are like DNA, RNA, etc., and those nitrogenous bases. And proteins, of course, the amino acids are the part that have the nitrogenous waste. So you have these amine groups that then have to be removed from the body. And there's three basic types of nitrogenous waste. Ammonia is a very common type. It's in most aquatic organisms, including some bony fish, and or most bony fish really, and most marine invertebrates, etc., will excrete ammonia. So this is in marine situations. Mammals and a lot of amphibians, sharks, and bony fish, they have urea. Now this has to do with being in either salt water situation, high salt water situation, or possibly high salt water, or in terrestrial situations. Urea is a little bit more of a stable compound, and then it can be easily taken out of the body. And reptiles and birds, insects, land snails, etc. Those guys use uric acid. It's a more complex sort of substance, but it's non-toxic to cells. So it's basically most likely evolved because of egg formation. That is amniotic eggs, because the waste products from the breakdown of proteins in amniotic eggs has to be non-toxic, since it can't go in and out of the eggshells. All right, so those are the 3 types of waste. The next thing is talking about the basic process. Okay, so the basic process involves 4 parts. Okay, 4 parts are filtration, reabsorption, secretion, and excretion. Now what is happening, this is a picture of a kind of a general nephron. That is one general filtration system. And it's going to have the red side there, this side right here is the blood vessel. And it is going to have blood that is coming in and then returning back. So the blood vessel is going to run along the filtration duct, okay? So what is happening is the blood will come in, and it has a lot of capillaries that will be sort of thin-walled and allow materials to pass via the wall of the capillary into the filtration duct. These are things that the body wants to get rid of. Mainly we're talking nitrogenous waste here, but other things will end up in there as well, like ions like calcium, for example, or sometimes like hydrogen ions, sugar, sometimes if there's excess sugar in the blood, or amino acids, those kinds of things. And those things, of course, the body doesn't really want to get rid of. What it really wants to get rid of is the nitrogenous waste. Okay, so the first thing that will happen is the filtration part. So the filtration part is just coming into the duct. So these are just molecules that would be entering. Of course, some water gets in there too, right? Because there's higher solute concentrations, so water will move into here. Now, the second part is what they call the reabsorption part. Now, this is, selective. So that means that there are things that can selectively be, the vessels or the sides of the duct are going to be selectively permeable to different types of solutes. The things that the body wants to reabsorb, like amino acids, for example, or magnesium or calcium or something like that. And it wants to reabsorb those things, and it will go ahead and reabsorb them. Sometimes it has to be actively transported, sometimes not, but it's still selective. And then there's secretion. That is where things that the body is trying to get rid of, or maybe has excess of, will pass the other way into the ducts. So this is not necessarily urea, but it could be. And then the very last thing is excretion, that is going out the duct. All right, so that's the basic process.